Good morning, everyone, and welcome to today's webinar, Simplifying Exosome Isolation with IZON. I'm Cheryl, the Marketing Manager at Atlantis Bioscience, and I am delighted to be your host for this informative session. Before we begin, let me briefly outline the agenda for today's webinar. So we will start off with a brief introduction to Atlantis Bioscience, followed by the presentation by Dr. Ong Ching Yi on the QEV system and how it simplifies exosome isolation. And finally, we will end with a live Q&A session. To ensure a smooth and productive webinar experience for all participants, I would like to touch upon a few online etiquette guidelines. Firstly, please keep yourself on mute during this entire presentation to minimize background noises. Secondly, feel free to submit questions through the Q&A box in the Zoom now at any time during the webinar. We will address as many questions as possible during the Q&A session. And as for those who are facing any form of technical issues, feel free to use the chat box to privately message me, or you can drop us an email at support@atlantisbioscience.com. So now, let's go in a little bit of an introduction about Atlantis Bioscience. We are a company dedicated to support researchers on their journey from bench to bedside, providing high-quality end-to-end solutions and services for several key research and development spaces, including cellular and molecular studies, drug delivery and development, cell and gene therapy, bioprocessing, and alternative protein. We are Southeast Asia's master distributor of BioWest, a premium cell culture reagent manufacturer from France. They are known for their low endotoxin level and high batch-to-batch consistency. We also offer a wide range of imaging dyes for different applications, such as exosome and EV labeling, qPCR, Western blot, flow antibodies, and more. As for drug delivery and development, we offer a range of specialized cell culture devices and microfluidics plates, preclinical mouse models, EV isolation and characterization instruments, as well as automated dissolution system. As for cell and gene therapy, this is where we have the most extensive portfolio, from specialized media for immune cells and stem cells to supplements like human platelet lysates and other important growth factors. We also recently expanded our portfolio to include viral vectors as well as mRNA lipid nanoparticle production. When you are ready to prepare for a scale-up, we also have the right solutions for you. So at Atlantis Bioscience, we carefully curate our product portfolio, selecting only trusted and proven brands from around the world. We are committed to ensuring that the products we distribute enhance research efficiency, improve experimental outcomes, and ultimately accelerate the development of life-changing therapies by you. Now, without further ado, I would like to introduce our esteemed speaker, Dr. Ong Ching Yi, our product expert for various of our Atlantis Bioscience solutions and services, including today's highlight, IZON Science. So let's welcome Dr. Ong.
Thank you, Cheryl. Thank you, everyone, for coming here today for our sharing session on IZON QEV Isolation and Characterization Platform. So before I begin, I would like to do a brief introduction on the extracellular vesicles, which we often call it EVs. Extracellular vesicles are lipid-bound vesicles secreted by the cells, which have the size of nanometer to micrometer. Various types of EVs are indicated based on their size and production pathways, including the exosomes, microvesicles, and apoptotic bodies. EVs, through their complex cargo, are able to reflect the states of their cells of origin, as well as to change the function or phenotype of other cells. Their vital roles in cellular communication makes EV a promising candidate for therapeutic agents such as vaccines, drug delivery system, disease biomarkers for diagnostic, and even in cosmeceutical and nutraceutical application. This figure shows an exponential increased trend of EV research, particularly based on the stem cells since 1991. The research focus of EVs are mainly on cancers, neurodegenerative disease, cardiovascular disease, and infectious disease. Other than stem cells, majority of the EV researchers is now focusing on collecting EVs from solid tissues as well as bodily fluids such as the blood, saliva, urine, and cerebrospinal fluid for both diagnostic and therapeutic. EV isolation of bacteria, plant, and milk also shows an increasing trend in various fields of research, thanks to the great potential of EVs. EV is holding so much clinical potential. However, the limitation in many isolation methods present major hurdles. There are various types of EV isolation method out there, which includes the precipitation, differential centrifugation, density gradient centrifugation, affinity-based method, filter concentration, size exclusion chromatography, and AF4. Among all, size exclusion chromatography, the SEC, is the principle adapted by IZON's QEV column. IZON's QEV isolation and measurement platform aims to bring standardized precision, customized separation, and high purity to your EV workflow. Let's look at this video to understand more on the QEV isolation principle of IZON column.
QEV is based on size exclusion chromatography. It works by separating extracellular vesicles based on their size as they pass through a column packed with a porous polysaccharide resin. As molecules enter the resin, smaller particles become trapped in the pores, delaying their exit from the column. As fraction volume is collected, particles are distributed across the volume based on size. Some void volume is eluted first, followed by the vesicles and then other smaller particles. This provides the most rapid, repeatable, and effective EV isolation system available. Additionally, with the automatic fraction collector, QEV is capable of being scaled up to thousands of samples.
This is a standardized workflow of EV isolation, where IZON products perfectly fit in every step. EVs from plasma, cell culture media, urine, and other biological fluid can be purified using QEV column manually or using the automatic fraction collector AFC. The EV isolates will then be characterized using the EXOID TRPS system to quantify the size, concentration, and zeta potential of your EV isolates. Downstream analysis, such as the proteomics and RNA analysis, can be proceeded using QEV concentration kit, which is optional, and also the RNA extraction kit. So, what's the reason of choosing SEC-based QEV isolation platform? Keeping EVs intact is very critical for functional studies. Ultracentrifugation method that uses high gravitational forces can impair the quality of isolated EVs, causing degradation, aggregation, and even fusion of EVs. While QEV isolation method only relies on the gravity without additional external forces applied during the separation. This allows you to study the intact EVs at their native form. Other than the excellent recovery rate, QEV column also offers superior purity. In most of the cases, purity will be the main concern. For diagnostic, we would like to have as little contaminant as possible in our samples so that the biomarkers will be easier to detect. For therapeutic purpose, we do not want to introduce contaminants such as the non-EV associated proteins into the patient. All of us know that standardization is very important in EV research. So, the use of differing techniques, varying protocols, and non-standardized equipments are not helping in this department. However, QEV columns coupled with the automation provides a more reproducible protocol through standardization, while also offering customization in the collection parameters. Last but not least, scalability of QEV columns can be achieved with the wide range of column sizes available to suit different sample volume for your research. We'll be looking into it in the next few slides. Milk-derived EVs are widely known because of their potential for immune enhancement as well as their potential in nanomedicine. This publication showcases the comparison of EV isolation method using several commercially available kits. Purity and the number of EV particles are widely recognized methods to measure the effectiveness of an EV isolation method. As we can see, IZON QEV column achieved the second highest purity, not much of a difference as compared to the immunoaffinity isolation using MAC capture. While the second bar charts shows that the IZON QEV column is able to yield more than 10 billion of EVs as compared to other methods. Let's look at this table for a summary. Ultracentrifugation and membrane affinity column did not show a good yield and purity in isolating milk EV. Additionally, the ultracentrifugation might damage or affect the functionality of EV to a certain extent. Precipitation was one of the early methods, where the polymers such as the PEG are used to form a mesh to trap EVs, allowing them to be pelleted along with the polymers. However, this method also traps the non-EV components, such as the protein, making it non-specific. Although it's a more gentle and faster method compared to ultracentrifugation, the presence of polymer complex with EVs may hinder downstream processes. Immunoaffinity isolation can indeed give you a high purity, but it also limits to the isolation of a subpopulation of EVs. In this scenario, the EV yield would be much lower as compared to the others. Although QEV and EV second L70 are SEC columns, are size exclusion chromatography columns, the obtained EV counts were different. This is mainly due to the pore size, filling, and the precipitation of columns, which will affect the resulting purity. In short, QEV column is highly recommended for those who aims to obtain a high yield of EV with significantly high purity. The QEV columns range spans a variety of sizes, all of which are available in three types: 20 nanometer series, 35 nanometer, and 70 nanometer series. The name of this QEV series derived from the pore size of the resin being used for the particular range of column. The resulting purified isolate would have slightly different characteristics as stated in this table. 70 nanometer series is recommended for those who are dealing with plasma or serum sample, as it has been proven to effectively remove up to 99% of the lipoprotein in the sample. 35 nanometer series is the most common type to isolate EVs from cell culture media. QEV columns are not restricted to isolate only EVs. If you are working with small viruses or other types of nanoparticles, you may also use QEV column to purify your samples. We offer a various column sizes optimized for different sample loading volume. The wide range of QEV columns make it relevant for all applications, from EV diagnostic and fundamental research to industrial processing for therapeutic and even cosmetic application. If you are exploring to scale up your EV production, you can opt for QEV 100, which allows the isolation of 100 ml volume, or you can even inquire for a customized volume that can accommodate up to 400 ml. IZON QEV columns are being used by global biopharmaceutical companies. Therefore, the option of GMP-grade columns is also available and can be produced under request to align with your regulatory standards. This is a typical elution profile of QEV column. The purified collection volume PCV can be optimized based on the elution profile of each sample and the downstream process. QEV column is well optimized to obtain an excellent separation between the EVs and non-EV component, which is the protein. You can see from this elution profile, the soluble protein only start to elute after the peak of EVs. If you are prioritizing the EV concentration, you can choose to collect the fractions that contains relatively high concentration of EVs. If EV purity is your main concern, you can collect earlier fraction before the protein starts seeping in to maximize the purity of your samples. In another scenario, where you are trying to maximize the EV recovery, you can choose to collect a larger volumes of fractions, but it comes at the cost of collecting volumes with a higher protein level. The purpose of collecting individual fractions is usually to determine the purified collection volume that you want to collect by observing at the fraction does the contaminant start to seep in. You can also identify which fraction elicit a better bioeffect for your functional studies. Other than collection individual fraction, you can also choose to select a single fraction of your desired volume after the optimization process. However, QEV column is an effective and efficient tool for EV isolation. They are just one part of the puzzle. Further efficiency and reproducibility can be achieved when the automatic fraction collector, AFC, kicks in. AFC automates EV isolation and enables precise, customizable, and reproducible separation. It is very suitable for small-scale and high-throughput processing of samples. Each QEV column is recognized by AFC via RFID tags, where it will remind you how many times are left for you to reuse the columns. Forget to mention that QEV columns can be reused up to 5 times for every single sizes. So you can customize the collection parameters such as the buffer volume and correction volume. Precise measurement of fraction by weighing every drop can be easily performed using AFC to ensure a high level of reproducibility. The automatic rotational carousel of AFC can accommodate both 2 ml and 5 ml tubes. Users can use multiple AFC side by side for parallel processing. IZON AFC is the world's first high-precision scalable EV isolation
is the world's first high-precision scalable EV isolation system. The AFC automates the previously manual QEV isolation process, which is based on size exclusion chromatography. Called smart columns, each QEV is equipped with an RFID tag, enabling the AFC to check what kind of column it is and record the number of times it's been used on the column itself. Precisely weighing each drop of your valuable sample, human error is no longer a factor. One user can also operate multiple systems in parallel. This is incredibly exciting for the EV field, as there's finally a repeatable system that can be scaled up for clinical trials.
It is true that AFC can help you in the automatic collection of fraction. But if you do not have so much samples to run and feel comfortable to run your samples using QEV columns manually, you can actually do so without using the AFC. This publication showcased the isolation of EVs from plasma and cerebrospinal fluid samples using the QEV original 70 nanometer column and the AFC. They are using a combined method of serial centrifugation to isolate medium EV fraction before subjecting the medium EV isolate to the SEC column for a purer yield. Besides that, an Amsterdam-based team working on EV miRNA as the diagnostic marker for lymphoma patients has recently reported their major progress in technical validation and establishing the QC steps to meet the regulatory standards towards IVDR compliance. They detail the quality control steps incorporated in their EV miRNA qRT-PCR assay using the QEV isolation and AFC as the method of choice for their experiment. All of this was achieved using a quality management system certified by ISO 13485. The implementation of QC steps enhance the reliability of the assay's diagnostic performance, advancing the quantitative measurements of EV mRNA towards clinical application. These are other publications that utilize QEV platform for the isolation of EVs from various sample sources, such as cucumbers, cerebrospinal fluid, bacteria, as well as red blood cell EV. For the EV characterization part, I'd like to introduce the exoid. The EXOD is a tunable resistive pulse sensing TRPS system which uses a single particle measurement technique designed for measuring nanoparticles. It is able to simultaneously measure the size and concentration, or size and zeta potential, with a level of precision and accuracy not found in commonly used light scattering techniques that provide only bulk estimates. When particles pass through the tunable pore, they are able to disrupt the electrical current in the pore, creating a blockade signal. The blockade magnitude, frequency, and duration are then used to calculate the particle size, concentration, and zeta potential respectively. TRPS technology is based upon the Coulter principle and the use of a tunable and stretchable nanopore. This works by analyzing the nanoparticles suspended in electrolytes as they pass through a nanopore. It is dependent on the transient changes in the flow of an ionic current, which gives you a graph on this figure. Nanopore can measure particles across a wide size range of 40 nanometer to 11 micrometer. Other than that, TRPS can resolve populations in multi-model samples. Unlike the dynamic light scattering method, TRPS measurements are independent of the particle or dispersion optical properties, allowing you to confidently measure the samples without being disturbed by the heterogeneous optical density. The impedance of a nanopore in an electrolyte fluid is sampled 50,000 times per second. As the particles are driven through the nanopore by applying a combination of pressure and voltage, each particle causes a resistive pulse or blockade signal that is detected and measured by the application software. Other than measuring EVs, researchers can utilize TRPS method to analyze liposomes or lipid nanoparticles for vaccine and therapeutic development, investigate the viral titer as the degree of infection, as well as to measure the bacteria in the samples. A lot of you will be asking, what's so good about TRPS system when it is compared to other systems out there? This is where I can show you the difference. This slide shows a comparison study of different nanoparticle analysis method published in the Journal of Extracellular Vesicles. Systematically measure the size, concentration, and particle size distribution of a quadri-model sample with similar ratio. This study involved tunable resistive pulse sensing TRPS, nanoparticle tracking analysis NTA, nanoflow cytometry nano-FCM, centrifugal liquid sedimentation CLS, asymmetrical flow field flow fractionation AF4, and also the multi-angle dynamic light scattering method MADLS. As we can see from this result, NTA analysis cannot resolve the mix sufficiently, while the TRPS shows a very good resolution of 4 peaks that represented the measured particle size distribution. Repeated measurements of nano-FCM are slightly size shifted and cannot resolve the last 2 peaks clearly. But the measurement CLS and AF4 are also able to measure 4 peaks, but the measurement wasn't accurate due to the size shift issue too. MADLS shows a very broad distribution without any feature. So in conclusion, TRPS is the best technique that allows you to properly resolve the peaks of 4 EVs in this system. Another recent publication showcased the accuracy of TRPS characterization as compared to the NTA analysis based on the cryo-TEM analysis. Spirulina EV in this study was concentrated and purified using QEV original 35 nanometer column, which were then subjected to characterization using 2 different methods, the NTA and TRPS. TRPS shows a more accurate data in terms of number and size distribution based on the actual one measured by cryo-TEM analysis, while NTA detected a population of EVs with a larger size range as compared to the actual one. Other than that, for those who wish to further concentrate your samples following the QEV isolation, we do offer the QEV concentration kit. Specialized nanotrap particles are used to convert the sample into pellet, which can be then reconstituted into any volume that suits your downstream application. The concentration kit is compatible for Western blotting, mass spec, and RNA analysis. We also have the magnetic version, which offers a quicker concentration step. IZON is currently improvising their QEV RNA extraction kit, which will be launched soon. Good news for the users who are looking into scale-up productions and high-throughput sample processing. New developments such as QEV Zenko, QEV Dexter, and TFF sample processing workflow from IZON is now launched. QEV Zenko is an automated chromatography system that helps user to run a larger sample volume. It comes with an inbuilt UV detector to monitor protein peaks and pH meter to detect cleaning processes before loading another sample. QEV Dexter is a system that's designed to process hundreds of samples per day. It is very suitable for high-throughput sample processing for diagnostic purpose. It can be customized to perform additional functions such as RNA extraction and other sample processing steps. When it comes to large-scale EV production, IZON QEV TFF system fits in the workflow perfectly by concentrating large volume of sample fluid efficiently before subjecting the concentrated samples to QEV system for the isolation of pure EV products. Apart from EV isolation and characterization solution, I would also like to briefly introduce you to the Biotium EV portfolio, where we offer different types of exobright EV membrane stains and exobright EV antibody conjugates. Unlike other membrane stains, such as the BKH, DIO, or DIL, which have a poorer solubility and can form aggregates that to be confused with EVs, Biotium developed the Exobright series to overcome these challenges. Some stains such as the CTB and WGA EV stains are even optimized for the staining of purified and bead-bound EVs from a broad range of sources. Lastly, our company also carries a comprehensive portfolio of cell culture reagent suitable for EV studies, as well as the specialized media for MSC, T cells, and K cells for translational studies. That's all my sharing for today. I hope my presentation can provide you a better understanding on how IZON's QEV platform can benefit in your research. Thank you.
Thank you, Dr. Ong, for the insightful presentation. I'm sure the audiences gain valuable knowledge on how to simplify exosome isolation with the QEV system, and I think they are all very well introduced to the advantages of IZON Science solution for EV's R&D as well as production. So now we will be answering some questions if there are any available. So far, I saw that we already have 4 questions here. So there's this person called John. He seems to be very interested in the IZON. He has 3 questions and one from an anonymous participant. So let's answer John's question. He is asking, how long does it take to run a sample?
Actually, the duration to run a sample using QEV columns depends on the types of columns that you choose as well as the column size itself. I can give you an example. For QEV original 35 nanometer series, it takes only 15 to 20 minutes to run the samples. This excludes the washing steps that is required in between samples.
Okay, it sounds like it's very short to process a sample. Sounds like timing is it's quite short. Then another question that we have from John is, what if I don't finish the 5-time use for one column? Can I save it for future users?
Yes, of course. If you are unable to finish up the 5-time use for one column, you can save up the column. You can store the column in a bacteriostatic agent, which is the 0.05% sodium azide, and keep it in room temperature for future use.
Okay,
The
so
shelf
you
life of
can.
the column is just 1 year, so we
Mhm.
recommend you to use it before the expiry date. Yes,
Okay, understand. So one column you can use it for 1 year as long as you store it properly. Okay.
correct.
So the last questions from John is that can the columns also be used to clean EV samples from fluorescent labels or antibodies? I think this is a very good question here.
Yeah, it's a very good question. Yes, the columns can also be used to clean the EV samples from the fluorescent label. For example, if you already stained the EVs and you wish to get rid of the non-binding fluorescent label or antibodies, you can definitely use our QEV columns to do so.
Okay. So that's good to hear that these QAV columns can avoid this. So okay, we have two anonymous questions here. Thank you, Jonya, for your questions. So here the anonymous participant asks, does the Exoid use laser to detect particles at all?
TRPS is an impedance-based system, so no laser is involved. Impedance means that it utilizes the opposition of electrical flow to measure the nanoparticles. So yeah, lasers is not involved.
Okay. So I hope that answers your questions. There's no laser involved. Then one more question here is, what are the packaging sizes for the columns?
The packaging sizes of the columns is also depends on the size of column of your choice.
Mm-hmm.
For QEV single, we
Mm-hmm.
have 20 columns per boxes. And for QEV original, we have 5 per box and so on. Yeah, if you wish to know what is the packaging size of your column of your choice, you can email us.
Okay, so for the anonymous participant, do email. We will let you know the email in when we send you the recording for Dr. Ong, so you can directly approach her to understand as well. Here we have one more last question here, unless there's more coming in, but at the moment there's this last question from Nitikan. She's asking, he or she is asking, could you explain more about scale up EV with TFF? Can this technique be used in clinical trial?
Okay, probably I can share screen for this particular question.
Okay.
Yeah, so we have this TFF system as a standard model as well as for cGMP environment. So for clinical trial, I believe that cGMP grade is needed. So yeah, for cGMP grade, we can customize the sample input up to 20 liter. So yes, it can be. I mean, we also offer cGMP grade for the TFF system.
Mm.
Mm.
So can this technique be used in clinical trial?
Yes, this technique can be used in clinical trial, provided you choose the cGMP grade TFF system. If you
Okay.
need more information, we can share you the specification of the TFF system later on.
Okay. So Nitikan, we will reach out to you for this. So you can we will send you an email. You can reply back to Dr. Ong to ask for more questions regarding this TFF system, the scale up one. So I think that's about it for the questions that we have for today. Once again, I would say we will close the Q&A session. So once again, thank you, Dr. Ong, for addressing all the questions the audiences have and for the introduction to IZON's EV solutions. So for the rest of you, you can connect with us on Facebook and LinkedIn to stay updated with the latest events and news. So I think we can pull up our QR code here for you guys to scan.
Okay, let me show.
Yeah. So for those that are based in Thailand, you can follow our Thai Facebook page. So it will be all in Thai. Then whereas for those that are English speakers, you can follow us on our LinkedIn, on LinkedIn Global. So you can scan this QR code to stay connected with us. So if you would like to learn more about the QAV system, the exoid system or explore more about Atlantis Bioscience range of cell and gene therapy solution, please visit our website at atlantisbioscience.com. We also have we publish good blogs every single week. So if you want to get more information about cell and gene therapy space, different lab tips, you can find it on our website. And we will be sending a recording of this webinar and a series of related resources to you through email as well. So do keep a lookout. And if you have any further questions and would like to discuss your specific research needs, please do not hesitate to contact us. So for those of you, if you are interested in AAV virus, we actually have a next webinar in February 26th, where we will cover efficient production for AAVs, so viral vectors. So those of you that are in the cell and gene therapy space interested to do that, you can follow us to get more information about that. So thank you all again for your time and attention. We hope you have a productive day ahead. Thank you and goodbye to everyone.